Hi, my name is Ashar Siddiqui, and I am a third-year Bachelor of Health Sciences student here at the University of Calgary. And the title of my research project is Predicting Lung Cancer Recurrence Using a Multiplexed Gene Signature. Lung cancer is the leading cause of cancer death in Canada, for both males and females. Lung cancer can be divided into two broad types: non-small cell lung cancer and small cell lung cancer. Non-small cell lung cancer can be further divided into three subtypes: adenocarcinoma, squamous cell carcinoma, and large cell carcinoma. The focus of my project is on adenocarcinoma and squamous cell carcinoma, which together account for 70% of all lung cancer cases. Treatment options vary for lung cancer patients and are based mainly on two factors: lung cancer type and lung cancer stage. For early stage non-small cell lung cancer, a common treatment option is curative surgery, after which the patient is given either adjuvant chemotherapy and/or radiotherapy as a way of preventing lung cancer recurrence. However, the issue is most of these patients that receive adjuvant chemotherapy or radiotherapy don't actually need it. So these patients receive all of the toxicities associated with chemotherapy or radiotherapy without receiving any of the benefits that chemotherapy or radiotherapy aim to provide. And so the purpose of this research project is to work towards developing a prognostic tool that can be used to predict the likelihood of a patient experiencing lung cancer recurrence after surgery. Therefore, in a clinical setting, this information can be used to assess whether or not a patient needs chemotherapy. Let's start with adenocarcinoma. In this study, 488 patients from the Cancer Genome Atlas, or TCGA, were used. From that, 325 patients, or 67% of the original cohort, were randomly selected to form the training set used in the study. After that, a machine learning algorithm was used to identify a list of 77 prognostic genes that correlate with patient survival. Let's take a look at a heat map. So in this heat map, red represents overexpression for a particular gene. So when we group all of the 325 patients based on their level of expression of those 77 prognostic genes, we get three distinct groups, as seen by the vertical bands on the heat map itself. Next, if we look at the corresponding Kaplan-Meier curve, or survival curve, we can see that the green group, which had the highest level of expression for these genes, has the worst prognosis, with a median survival of only about 20 months. These would be the types of patients in this group that would receive chemotherapy after surgery. The highest median survival we see in this Kaplan-Meier curve is with the red group at 55 months. Now, to really find a group with extraordinary survival, this study took on a unique two-step approach, which means that we repeat these steps, except that we only look at the patients in the good prognostic groups, so the red and blue groups. After finding a new set of prognostic genes, here's the heat map for step 2. As we can see, we have the blue and red labeled prognostic groups. And when we look at its corresponding Kaplan-Meier curve, the blue group has an extraordinarily high median survival at 106 months. This is the type of group that may only need less severe forms of chemotherapy, or they may even be able to avoid it altogether. The same methods were used for squamous cell carcinoma, which involved using 474 patients from the Cancer Genome Atlas, of which 316 formed the training set. Here are the results of the first part. The red and blue groups had median survival of 71 and 20 months respectively. From here, a second set of analyses was run on only the red group. And here are those results. As we can see, the red group has extraordinary prognosis with a median survival of 108 months. Therefore, so far in this project, we have identified unique gene signatures through which we are able to predict the prognoses of lung cancer patients after surgery. After creating a prediction model and validating these results, the results shown here can be used to develop a prognostic tool for clinical use. The overarching goal of this prognostic tool is to move away from a one-size-fits-all approach and towards an individualized approach. By tailoring treatments to each cancer patient individually, we can provide them with the correct level of adjuvant chemotherapy or radiotherapy that they need, and we can avoid overtreatment and all of the unnecessary toxicity associated with it.